So welcome everybody. This is a somewhat unconventional back to league meeting. As I'm sure you already know, we have two really excellent speakers tonight who are going to talk to us about COVID-19 and its local impacts. But first, I have a couple of updates. I'll talk about the National Convention, and then Judy, I'll let you hop in and tell us about the Observer Corps. The National Convention went really great this year. Obviously, it was all online, so it was a little bit different, and there were definitely some hitches, but I thought they did a really great job getting it all put together. We had 1,267 delegates from all 50 states plus DC and the Virgin Islands. We also elected our new president, Dr. Deborah Turner, who is our 20th league president nationally, obviously. I'll go over a couple of notes from which votes went where, but I'm gonna post a link to the chat right now because they did a really good job briefing on the convention, and it has all of the vote counts for everything that we voted on and a summary of all the speakers. So I definitely encourage you to give a read through that if you want to know all the details on what happened at National. But we approved the budget, we kept the per member payment at $32 per person, and we agreed that students will continue to be free in terms of dues indefinitely, so that's added to the bylaws. We added abolishing the Electoral College to our biennium program for 2020-2022. We adopted the concurrence on electoral systems in a landslide of 1,064 to 79, which I was really happy to hear about, so that's why I included it. We also reaffirmed our commitment to immigration reform and updated several other positions. And again, that's where the full summary is. I've posted the link, so hopefully you can go and read through it. It's a really good summary. So that I will hand it over to Judy to tell us a little bit about what Observer Corps has been up to.
The Observer Corps, can you hear me?
Yes.
Yes.
Okay. The Observer Corps has been busy all summer long. People have done a phenomenal job of attending the virtual meetings and reporting on them very quickly. After the August library board meeting, the Observer was, well, at the meeting, the Observer heard a request by the Pullman Library Board for letters of support that they could send to the mayor and to council members supporting the library, specifically for funding requests in the very near future. And so the league did that. Bobby, I'm not sure who actually wrote the letter, but it, as always, goes out under Bobby's name and was sent out on August 18th. So we heard that they very much appreciated that support. And I really don't have anything else. That's it.
Okay. It was drafted by Libby. The letter was drafted by Libby.
Okay.
Awesome work, Libby. All right, so the last announcement that I have before I introduce our speakers, I'd like to remind all of our members that dues are due. It's still $65 for individuals, $90 for families, and $3 for students. However, if you're a new member who's joined us after January 1st, that doesn't apply to you. You're perfectly good on dues, and also welcome. So that's announcements are done. So without any further ado, I will.
Okay, wait a minute. Can we pay? We can pay our dues
without further ado on the website.
On the website.
Perfect. Good. It's easier than ever.
Or you can send it by regular mail. It's a couple of dollars cheaper if you do that.
Good point.
To either the league address or to my address, Paul Spencer.
Awesome.
Don't forget the nonpartisan statement, Helen.
Oh, you're right. So the league is a nonpartisan organization, which means that we do not support parties or candidates. We do encourage and appreciate the, I haven't done the partisan statement before. Okay, we do support and encourage the value that's brought by our members having had experience in elected office, but our board members are not, like myself and Bobby, not allowed to participate in elected office or political campaigns while we are, on the board. So that's the nonpartisan statement. Did I miss anything, Libby?
Well, we do take positions on issues after
We
careful
do
study.
take positions on issues. That's correct. No
You
candidates, no parties, yes issues.
We mentioned immigration reform as one that came to the forefront at the National Convention.
True, true. And there is debate within the league over which issues we should take positions on for sure, but we do that. Yes.
Okay,
Okay.
good.
Now, without any further ado, I will introduce our speakers. I know the tension is so high. So first, we're going to have Dr. Bonnie Gunn, who is a virologist with the School of Global Animal Health, Paul G. Allen. And she is going to tell us about viruses and COVID-19 in particular. So take it away, Bonnie.
Okay. All right. Thank you so much. So can you all hear me?
Yes.
I hope so.
Yes.
Okay, fantastic. All right. So I'm going to share my screen and I've got a little presentation that I've prepared for you all. So as Helen mentioned, my name is Bonnie Gunn, and I am actually a relatively new assistant professor in the School of Global Animal Health at Washington State University. And my focus has primarily been virology and immunology. So it's a real pleasure to talk to you today sort of about SARS-CoV-2 and kind of give you a little bit of my perspective on sort of like at least what we understand. I do want to say that this is still a constantly evolving situation. So you know, things are changing all the time the more we learn, but it is incredible just how much we've learned in the short time that the pandemic has been a big part of our lives. Okay, so first I just wanted to go over sort of a general overview of kind of virology and how viruses replicate, and really starting with the basic question of really what is a virus. And so at its basic level, viruses are encapsulated infectious particles that contain genetic information. And this can really be DNA or RNA, which are the building blocks pretty much of life. And the second requirement for a virus is that they do require a host cell to replicate. So there are millions of different viruses around. Many of them actually do not cause any disease. Many of them we don't even know about at the moment. But there are, of course, quite a few that have emerged into the human population and cause disease that we are now very familiar with. So this can be something that we experience every year, such as influenza, lifelong infections such as HIV and herpes viruses, as well as these sort of pandemic viruses that come out. So this includes coronavirus, and a topic that I've studied quite a bit in the past is Ebola virus. And so as I mentioned, viruses do need a host cell to replicate, and this doesn't necessarily have to be a mammalian cell or a vertebrate cell. And virtually all living organisms on the planet can be infected by viruses. So this includes humans, bats, livestock, invertebrates such as ticks and mosquitoes, rodents, even marine mammals. So there's a whole whale virome that is being studied at the moment. And even bacteria can be infected by viruses as well. And there's this little red guy in the middle here called a bacteriophage, which is responsible for that infection. And so kind of going along the lines of that viruses need to replicate inside a host cell, one of the major barriers for viruses is actually to get into a host cell. And so most viruses actually do this through binding a protein receptor on the surface of a host cell. And here I've used coronavirus as an example where the spike protein of the virus has to bind to a receptor that allows it to internalize into the cell. So there are only, some receptors. So these receptors can be very specific for certain viruses, and some receptors are only expressed on certain types of cells. And so this is what we call tropism. So if a cell does not express the receptor that a virus needs, it cannot infect that cell. And so the receptor for SARS-CoV-2 and the original SARS, which I think we're now calling SARS-CoV-1, is a protein called ACE2, which is not on every cell, and it's expressed on specific cells in the lung and in the gut. And now we are actually also discovering that it can be upregulated in different disease states as well. So we're sort of learning more and more about the expression of this particular receptor on all of our cells, which then helps us understand why the virus can target certain organs or not. And so once a virus, so I'll just give a sort of an overview of like what would happen in a single cell upon viral replication. And so the first I've mentioned is already sort of this binding to a host receptor and entry into a host cell. And then the next thing that the virus does is that it has to release its viral genetic material. So this is in the context of an RNA virus, such as coronavirus. So it releases its RNA into our cells, and then it starts hijacking our own cellular machinery to start producing more of itself. And so it uses the host ribosome here to make more of its own RNA, so more of its own genetic material. And then it still uses our machinery to basically to produce those proteins in something that we call translation, so that it can then start to make all of its different viral proteins that it needs either to evade an immune response or to make more of itself. And so this is where we have, you have the genome that sort of is constantly replicating, and then the various proteins of the virus package that genome back up, make a virus, and then we have what's called egress, where then the virus then escapes from the host cell to go on to infect a new cell, new tissues, and then can disseminate throughout the body. And so coronaviruses is the focus of tonight's talk, and of course the focus of many of us these days. And so coronaviruses have been known actually for quite a while, but they are one of the larger viruses in terms for genome size. And they were named primarily because of, when you look at them on a electron micrograph, as you can see here on the left, there's almost like this little crown around the virus, which was so Latin for crown is corona, so that was called coronaviruses. And what this crown really is actually the spike proteins on the surface of the virus. And so the spike protein in the viral membrane encapsulate this genetic material, the viral genome in here. And this is sort of the general structure for all coronaviruses and for many other RNA viruses as well, where you have protein on the outside, and then you have your genetic material on the inside. And so coronaviruses are, again, we've known about them for a little while now, but what's really kind of brought them to the forefront, even before COVID-19, was the emergence of two other viruses into the human population that had pretty devastating effects. So the first, of course, was the original SARS coronavirus, which we now call SARS-1, which emerged in 2003, and it roughly had about a 10% mortality, which was actually relatively high. And then in 2014, we had another outbreak of a Middle Eastern Respiratory Syndrome coronavirus, or MERS coronavirus, and that actually caused even more mortality, so about 30%. And so of course now in 2019, we have SARS-CoV-2, and right now it's looking like the mortality is at around 3%. So quite a bit less than the other coronaviruses, but it has infected, of course, many, many, many more people. And so the coronaviruses, of course, don't only infect humans, but they also infect animals. And they actually infect a wide range of animals. And so we have, there are about 3 different groups. So coronaviruses as a family can be grouped into 3 different families, really. So beta coronavirus, alpha coronaviruses, and gamma coronaviruses. And the ones that are circled in red are the viruses that are known to infect humans. And so these include MERS, these other viruses here that cause the common cold, and of course SARS, and now we can insert SARS-CoV-2 pretty much next to the original SARS. But the other viruses that have been sequenced have been sequenced from bats, actually. So there's, pretty much in every family of the coronaviruses, there are bat viruses that have been isolated, some that have been very, very close in genetic sequence to the SARS coronaviruses, or to SARS itself. But then there are also others. So MERS, we know, replicates in camels. There's a bovine coronavirus, mouse coronavirus, ones that also infect cats, pigs. Of course, there's like civet cats and pangolins and probably many, many more, that the more that we learn and sequence and look at our different sort of animal populations, the more that we're likely to find. And so one of the interesting things that, or one of the key questions I think that a lot of people have been asking is, sort of where did SARS come from? Or certainly where does SARS-CoV-2 come from? And so genetic analysis of different viral sequences have really started to shed some light on this question. And one of the interesting articles that has come out recently has been from a group of scientists who have been, again, studying these different genetic sequences from bats around the world, as well as different animals, and comparing the sequence to SARS-CoV-2. And what they found is that it looks like the lineage of virus that has given rise to the currently circulating SARS-CoV-2 has likely been circulating unnoticed in bats for decades. So this didn't exactly come out of nowhere, but it has been sort of probably testing the waters in a sense, sort of constantly coming into contact with humans, adapting to the point where it can then expand into the human population. This is undoubtedly not the only virus that is doing this at this point, so it's sort of a cautionary tale for what may happen in the future. So viral sequencing has also, even sort of beyond just looking at the origins of the virus, we can use viral sequencing to really track transmission. Sort of the group of scientists who have been doing like some absolutely fantastic work is actually the group that has been putting all their data onto this website called NextStrain. And this is a group actually out of Fred Hutch in Seattle. So sort of our neighbors have been really on the forefront of a lot of this analysis. And so by tracking the genetic mutations in the different viruses across, that have been isolated from different people, they can create this sort of transmission network where we can actually see, we can see whether a virus was transmitted from, let's say, California to somebody in New York, or New York vice versa, just based on the sequence itself. So this has really given us sort of an insight, not only, it's sort of insight into understanding the transmission patterns, especially, globally. But then you can also go on a kind of a finer scale and sort of understand, perhaps the particular events that people attended, or, whatever contact they might have had, to try and understand a little bit better about how this virus can actually transmit. So one of the major questions that I will try and sort of give you a little bit of an overview for, but of course we don't fully understand the real answer here, but is the overarching question of how do coronaviruses actually cause disease? And so this is sort of the part where I really would like to talk to you about the immune system and the role of the immune system in both protection as well as making disease worse in terms for some of these infections. So what normally happens in a viral infection, or any kind of infection for that matter, you have these immune cells, which we call sort of these sentinel cells. And these are cells called macrophage, monocytes, and dendritic cells. And what these cells do is they patrol around your body. And so when they come into contact with something that they don't recognize, then they internalize it, and then they call and they let other immune cells know. So they're calling in the cavalry, basically. And then this brings in other innate immune cells that can then come in and either take care of the pathogen, or just basically help destroy it and help localize, keep the pathogen in check. So at the same time, these sentinel cells can also start activating these very specialized cells called T and B cells. And then these are the cells that will actually create this sort of lifelong immunity, or this long-term immunity that we all hope for. And they do this through the production of antibodies, as well as these what we call memory killer T cells that can then rapidly expand once they see this pathogen in the future. And so ultimately, what normally happens, or what would happen in an ideal situation, is that you have this clearance of infection when the immune system is pretty much working as it should, clears out the pathogen without really, without too much trouble. However, we also have something that's called immunopathology, and many times this is really driven by this cavalry, the innate immune cells that come in. And so in some cases, these innate immune cells are coming in and they're a little too aggressive, a little too active, and this is really causing immune pathology. And ultimately, while you still may actually clear the infection, you also do a lot of damage to the host. And so this is where we can see severe disease coming up in some infections. And so one thing that has become very clear is that severe COVID-19 is associated with this immunopathology, or this overactivation of the immune system. And this is particularly bad and very devastating just because of the organs that it is impacting, which is the lungs. And so of course now when you have a coronavirus infection, you have infection of the lung tissues and the cells that are lining the lungs. And some people actually are able to clear. But if the virus gets down into these lower airway tracts, then we start seeing what's called a cytokine storm, and we start seeing this overactivation in immunopathology. So the virus will then activate some of those sentinel cells who send out something called cytokines, which are these little molecules that then basically call in the cavalry. So the cavalry comes in, they then see the virus, they want more reinforcements, they're sending out more cytokines, and then ultimately then you're having this sort of breakdown in these tissues, which then is leaking fluid into the lungs. And this is, of course, where we start having this severe respiratory distress. And this can have varying levels, as we've seen. You can have moderate damage where you still have some restriction, all the way to severe where you have very limited gas exchange in these tissues. All right, so we're sort of still, of course, like kind of understanding it. And, you know, as scientists, as we start developing better animal models and understanding a little bit more and studying more patients, then I think we'll have a better understanding of exactly what, you know, causes all these different differences in disease severity, and maybe why some patients develop severe disease while others do not. So then I just want to get to like sort of why, you know, this is a sort of a question and a topic that has kind of been brought up a lot of really how SARS-CoV-2 compares with other respiratory viruses, and really what makes SARS-CoV-2 different from those viruses. And so just looking first at the epidemiology of SARS-CoV-2, here called COVID, compared to the other respiratory infections, so flu, as well as SARS and MERS. And so we can see here that the case fatality rate for flu is actually anywhere between 0.05 to 0.1%. But of course, when you have that on a scale of millions of millions of people, then that, of course, that number is actually substantial. COVID-19, our current case fatality rate is about 3.3%, whereas SARS was, you know, up to 11%, and MERS was actually almost, you know, 34% case fatality rate. So the incubation is different across these different viruses. So influenza has probably the shortest incubation time before you start showing viral replication and symptoms, whereas the coronaviruses are a little bit delayed. You can also see that the infectious, so the R0, which is sort of the measure of transmission, is different between these viruses as well, where SARS is probably a little bit higher than COVID, but COVID definitely is a very transmissible virus, particularly compared to MERS or influenza. So there's varying levels of hospitalization. So with SARS and MERS, most of the cases that came up positive did require hospitalization, whereas right now only about 20% of COVID patients require hospitalization and 2% of influenza. And then the attack rate in the community, you know, it sort of varies as well. So in terms of really comparing SARS-CoV-2 with the other coronaviruses, there's two things that really have come to mind. And so the first is sort of the tropism of where the virus actually infects. And so what we are, so early on, I think clinicians were able to identify that there was a high viral load, which meant there was a lot of virus in the upper airways of COVID patients. And so this is probably, you know, so this sort of allows the virus to replicate sort of in these areas where, you know, you might have a runny nose. It's easier to touch your face and then touch something else. Whereas MERS and SARS, they were actually always replicating, and the highest viral load was actually in the lower airways. And so this would get expelled out if you were coughing or I guess mostly coughing, because you're sneezing, you could also sort of have more from the upper airways. And so even though SARS, SARS the original and SARS-CoV-2 sort of have this, they bind to the same receptor, so they in theory they could bind to the same types of cells, but it does seem that there is a difference between their ability to bind that same receptor. So SARS-CoV-2 looks like it can actually bind to the receptor ACE2 better and faster than the original SARS-1. So that might explain why, if a virus is getting exposed to your epithelium, and if it comes across a cell that is expressing ACE2, SARS-CoV-2 can actually bind to that cell better, get in faster, and actually start replicating, whereas SARS might not have the SARS original might not have the binding affinity needed until it gets all the way down into the lower airways. And so this, also might suggest, although it hasn't necessarily been proven, that there is faster viral entry, but it does look like SARS-CoV-2 does have some additional sites that are likely to enable it to enter into cells faster. And so another thing that has also become very, very clear about a big difference between the transmission of SARS-CoV-2 as well as the original SARS, and that's in terms for viral shedding. So viral shedding means I guess the amount of virus that is shed out into out of host cells that can then go in and infect somebody else. And so what has been clear is that when COVID patients are coming in and they're actually showing symptoms, their viral load from at least the nasal passages are declining sort of as their symptoms continue. And so then it actually looks like what is happening is that you have an increased viral load here before you ever start to show any kind of symptoms. But of course, most people don't know that they'll be infected or don't suspect that they might be infected until symptoms have come on. So this is in contrast to the original SARS, where the peak viral shedding didn't actually happen until over a week after somebody had started showing symptoms. And so what's most likely has happened during the SARS, the original SARS epidemic in 2003, was that once people started showing symptoms, they could be isolated. And then then they were completely isolated at the time where they were the most infectious. Whereas right now, of course, we have a lot of people who are likely shedding virus, infecting others before they even show any kind of symptoms. And there's also a growing appreciation for the fact that some people never develop symptoms, yet they still might be infectious. And children, for reasons that are still not well understood at all, are infected, but then they don't show symptoms as well. So there was a growing interest in trying to understand, what sort of is the contribution of these different populations to transmission. This obviously has a profound impact on how we sort of go about being able to stop this pandemic. Sorry if I'm going a little long, but I'll finish up soon. So one thing that has also been very clear is that there is sort of transmission by this aerosolization of droplets, and of course sneezing was one of the big, sneezing and coughing is sort of one big way that you can expel a lot of droplets. And there's been these very interesting studies of really trying to understand how far different droplets can actually go. And so in 2016, there was this high-speed imaging taken of somebody sneezing, and you could actually visualize the particles and how far they went. And at the time, within like a third of a second, these particles were going about 2.5 feet from the person who sneezed. And so there's new data that is now suggesting that with the differential impacts of humidity, temperature, and airflow, droplets can actually travel about 7 to 8 meters. So this is actually substantially larger or further than we had previously thought. Okay, so now I just want to turn to sort of the vaccines and therapeutics that are being developed right now. And I just want to sort of give a sort of explanation as to kind of what the differences are. So a vaccine, of course, would be given before you ever encounter a pathogen. And really what it serves to do is to really help train your immune system to recognize the infection or recognize the pathogen and then stop that infection. And so that would be through the production of antibodies or the induction of those killer T cells that I had mentioned earlier. And a therapeutic is really given after somebody has been exposed or infected to really help them fight the infection. And so the therapeutics that are being developed right now, they're they're taking a couple of different forms, and I'll go through a couple of them. So the first is to really try and target the ACE2 receptors. And so that's the receptor that is required for that the virus uses to get in. And so there's a whole group of people that have been working on making these sort of almost like just like mimics of the virus. And so they will basically bind to the ACE2 receptor, so to kind of like take up the receptor so the virus can't bind. And these are called these competing kind of receptors here. Another is to use antibodies. And this can either be in the form of natural antibodies, such as convalescent plasma. I know there's been a lot of talk recently about whether or not these might be efficacious for treatment of people, but there's also a growing body of work that really suggests that you can make antibodies in the laboratory that have been sort of optimized for the most effective activity against virus. And this is actually something that I personally work on. Where we try and find antibodies and make them better so that they can actually bind.
Can't hear.
That might be on her end. Dr. Gunn, can you hear us?
She's frozen.
Oh no. Hopefully this clears up in a moment.
Candidate.
There we go. We lost you for a second, but you're back now.
Okay. Whoops. Yeah, so that ultimately targets various parts of viral entry. So here it actually inhibits viral replication, or the RNA replication sort of early after viral entry. So another growing class of therapeutics is not necessarily treating the virus, but it's treating the disease. And so really targeting that immunopathology that I mentioned earlier. And so there have been there's actually lots of different trials that are going on right now. One of the early therapeutics that was of interest to a lot of clinicians and scientists was this one called tocilizumab. And this was basically inhibiting some of those cytokines and inhibiting that cytokine storm that would then serve to limit inflammation. And so one of the, I think steroids was just recently advised or recently promoted by the WHO as a possible treatment for severe COVID as well. And so that would also have a similar effect. It would dampen inflammation. And so finally, I just want to turn to vaccines. And so the vaccine development against SARS-CoV-2 has just been really quite impressive. I mean, we've gone basically 6 months and there's already 47 different vaccines that are currently in human trials, which is just sort of unprecedented in the community. And so there are several different vaccine strategies that are being explored right now. So there is an mRNA-based vaccine, and there's one that has like sort of it basically injects DNA into you, so you're producing your own protein producing your own viral protein that the immune system can recognize. There's ones that actually use other viruses, such as this adenoviral vector. And ultimately what all of these are doing is basically trying to give your immune system the bit of the virus that it needs to learn to recognize without actually being infected by the virus itself. And so that's the sort of strategy going on with these three. And there's also the more traditional strategy where you actually just take the virus itself and then you inactivate it and then give it to somebody. And so this is actually several of our most successful vaccines have used this approach. But the one that looks like it is kind of furthest along in the clinical trials is this mRNA-based vaccine, and I just want to talk about it just very briefly. And so right now, this is the one that has been furthest in clinical trials. And so this is a little bit of the data from the first clinical trial, where they had given study participants different doses of the vaccine. And so here we're just looking at this sort of antibody titer. And so this is called a neutralization assay. And so what we're looking for here is the ability of antibodies to neutralize and prevent the virus from entering into host cells, so pretty much blocking that interaction with the ACE2 receptor. So prior studies have sort of established that you need a titer of about 1 to 160, and so that's what this red line here represents. And you can see that by day 43, so after the vaccination, all of these participants here were able to induce antibody titers of that quality, so that's actually really, really promising. And there's also been animal challenge studies where you can vaccinate animals and then challenge them to see the efficacy of the vaccine. And this vaccine actually does look incredibly promising, where the vaccinated animals had accelerated clearance from the virus from the nose. So there's kind of lots of things that are constantly developing at the moment. I mean, these are the ones that are sort of being pushed at the moment. We will see whether or not it actually pans out in the larger community. But it is certainly an interesting and challenging time, I think, to be a virologist. Okay, so anyway, so I think that's all I have for tonight, but I welcome any questions or comments that you might have.
Thank you so much. That was a really enlightening presentation. Do we want to, I don't know how we normally do this, do both presentations and then one question section, or questions and then another presentation and then more questions?
I think we should go to the second speaker, Helen.
Awesome. Okay, so just hold on to your questions, hang on to them, and I will introduce our next speaker, and then we can have all of the questions at once. So Troy Henderson is the director of the Whitman County Department of Public Health, and he's going to be talking about the local demographic COVID data and also the plan for infection control in the county. So with that, I will let you take it away.
Thank you. I really enjoyed the previous presentation. I know I learned a lot from that. Most of what I'm going to say, most of you have probably heard or are familiar with in various press releases and other medium, but we'll do a quick summary. Basically, we had our first case, I'm pretty sure it was March 22nd, it was a Saturday. And then for the next 5 months, up until about August 20, we had about 175 cases. So we were doing pretty good for the first 5 months of this. We had a pretty slow rate. And it seemed like when cases cropped up, we were able to squelch it pretty quick with case investigation, contact tracing. And as everyone is aware, since about August 22nd, we've had about, now we're at, we've had about 500 cases in the last, 13, 14 days. And over 95% of those have been in an 18 to 23-year-old demographic in Pullman. I don't know the specifics of what percentage of those are associated with WSU or not, but obviously the vast majority of 18 to 23-year-olds in Pullman are associated with WSU. And as I'm sure everyone's aware, about 12,000 students started coming back in the middle of August for various reasons. I think the reason that's in the social media most frequently is the fact that folks had signed up for leases and felt a financial obligation. If they were going to have to pay rent, they might as well be here. I think there's also a component of COVID-19 fatigue, and folks had to leave early last semester, and folks want to socialize with their peer group. And so I think there's a number of reasons why people came back to Pullman. And when they did come back, they did indeed socialize with their peer group in collections large and small. And so as the presentation just showed, this virus has a pretty good R naught value, and if you get in pretty big groups and you're not social distancing and wearing masks, it spreads pretty effectively. And we've gotten a close-up view of how effectively it can spread in an environment that is conducive to that. So that
Troy,
gets us,
can you possibly speak up?
I can. I'm sorry.
That's okay. Thank you.
Yep. And so that gets us to where we are today. We've been having about 40 cases a day for the last 12 or 13 days. We've had a positivity rate. A couple of days ago, it was calculated at 16%. Anything that's above 5% suggests that you're not testing enough. And so additional testing has been brought on board, mostly in and around WSU and mostly led by WSU. The Range Health Mobile Clinic arrived first yesterday. Cougar Wellness Clinic started testing, I believe, Tuesday, maybe Monday, and are ramping up their testing activity outside at a little tent type area. And then, as I think most everyone's aware, there was a group of Army National Guard folks who are funded by FEMA who arrived today to look at different sites where they can set up, and they're going to set up and start testing on Tuesday. And then also WSU, the athletic, the medical folks who take care of the athletes with WSU have done another round of kind of surveillance testing with the athletes. And so there's been a fair amount of additional testing that has come to bear and is in the process of coming in and around the WSU area. And of course, the goal is to get the positivity rate below 5% so that case investigation contact tracing can be more effective and we can have a clearer picture as far as the accurate numbers as what's going on in our community. From the county public health standpoint, our biggest concern has been from day one and continues to be to today, protecting our most vulnerable folks in the community. And primarily that generally takes the way that happens is we protect long-term care assisted living facilities. That's a congregation of folks who are vulnerable that in the early stages of this pandemic have had some very negative outcomes when this virus has gotten in those facilities. And so the current outbreak that's occurring right now, because it's in such a narrow demographic in the health department, we feel as though the threat to long-term care assisted living facilities and other vulnerable people is kind of a two-step process. We think it will go from the 18 to 23-year-old demographic to the broader community. And then if it starts spreading in the broader community, that makes it more likely to impact individuals who have high-risk conditions or facilities that have a number of folks that meet that criteria. And so every day I get summaries of every case that are not WSU related. And so we watch very closely on a daily basis for any signs that it is going into the broader community. And we're about 12 days into this, and we have not really seen that. We've had about 12 cases that were not in that 18 to 23-year-old demographic, and the bulk of those have not been associated with WSU students. It was kind of that normal activity we were seeing beforehand. So I would suggest that before local entities take action to increase social distancing and restrict movement among folks in Pullman, and the local agencies that can do that are the County Board of Health and the County Health Officer. They also are watching to see if it leaves this narrow demographic that it's currently in. With that being said, we're also trying to address the outbreak that's in that demographic, primarily through increased testing, greater messaging to that specific age demographic that's being led by WSU and University of Idaho, that messaging program. And then we continue to do very aggressive case investigation, contact tracing. We have gotten to every case in less than 24 hours for case investigation and contact tracing, with the exception of folks who didn't answer their phone because they dropped it in the swimming pool or something. But we have sufficient case investigation, contact tracing to deal with probably up to about 85 cases a day. And we're expanding that capacity now. We anticipate next week when the Army National Guard team starts testing, we are likely to see an increase in case count because we're going to be doing more testing. And obviously with a 16% positive rate that we're seeing now, we're missing a number of folks that we hope to be catching next week. Absent a vaccine or a therapeutic treatment, identifying who has the virus and doing case investigation, contact tracing, and then quarantine and isolation is the most effective public health intervention that we have. And it's a public health intervention that has been in place for well over 250 years. And we do that very well in Whitman County. So I think that may be it for me for my update, and I'll stand by for your questions.
Awesome. Thank you so much. So I guess we will open up the floor to questions for either Dr. Gunn or Troy Henderson.
The question that I have is, relates to Moscow versus Pullman. I mean, what are the numbers over in Moscow? I can't believe they can be all that different.
They're very different.
Is it because they're not testing? Because they're not reporting? What's going on?
They are definitely testing their student body. The percentage of their student body that they have tested is probably higher than ours. Within their community, I don't know if their testing is as effective as ours is. But I have recognized that same difference, and I'm not sure what it is. I'm not sure if, of course, University of Idaho meeting in session is a smaller number of folks than WSU not meeting in session. But with that being said, there does seem a suggestion that there's a behavioral difference between the two populations, which you know, you might need a behavioral psychologist to address that.
I don't know if anyone knows about this, but I was also under the impression that they were testing preemptively, testing students before they came back to school. I don't know if that would have any effect on that either.
You would think if they're testing folks before they come back, anybody who would test positive then wouldn't come back. They would have been asked to quarantine and isolate, which would have an impact. But I'm not familiar with if they did that or how widespread it was if they did do it. I do see a question up here that says they've heard that public health in Idaho does not share data with Whitman, and that is not true. We coordinate quite well with case investigation contact tracing because, as you would imagine, there's folks who work in Pullman, live in Latah, or vice versa. And we have good communications back and forth across the border specific to tracking down folks who are positive and their close contacts. In other areas, communication could be better. Is there monitoring to be sure positive cases are isolating? No, not really. I do get calls from folks who think they saw their neighbor that they think is positive who's not isolating. Right now, we would not have the capacity to do that, and we don't do that. And part of the reason is because of the nature of COVID-19, the R0 value is not high enough and the severity of the disease is not high enough that you need to go beyond relying on individuals to do what they're asked to do. We recognize that 100% of them will not do that, but you don't have to impact 100% of the cases to drive the R0 below 1 and control the disease. And so that is driven largely by the nature of the disease, how rigorous you follow up with individual cases.
I have a question. It seems to me that we had enough at least news research given to us from the Ohio State University and from a number of schools on the East Coast talking about young people behavior, that we should have met these students at the Whitman County line and tested everyone and not be 2 weeks late. What happened to WSU with that timeline?
Well, the fact that WSU probably could have done more testing sooner, I'm not going to argue with, and I'm not a representative of WSU, but in regards to meeting people at the county line and testing, that's generally not something in the United States that is easy to do or will go over well before you're sued on various liberty issues. I do know that some indigenous nations that are independent nations have done that, but obviously they're in a legal position that's very different than a county government.
And I was stretching that, but it just seems to me we had enough information that we should not have trusted our students once they got here to provide the very best testing model that was available. Well, not available, because obviously it wasn't available, but nothing with Whitman County. I'm just wondering why WSU and its resources were so slow.
Yeah, I will say the fact that this age demographic brought virus and increased the viral load did not catch anybody by surprise. This is something we've been talking about since they left on spring break. I will tell you the one thing that I think caught all of us by surprise is the number that returned. So when they went to classes online, we did say, hey, there's going to be a chunk of them that are still going to come. But in those meetings and debates, I don't remember anybody saying they thought it was going to be 12,000 of them. So that number is higher than most folks would have anticipated. But the you're right that the anticipation that there would be greater viral activity, that did not catch anybody off guard.
Mhm.
And
Thank
to
you.
that end, we were ramping up case investigation contact tracing teams starting 4 months ago, specifically for this reason. And we had ramped up to a capacity to do 100 cases a day. So that was what we were planning for. And so in that regards, we've been under what we've planned for, but again, more of them came than I think most people would have thought. There's a question here about the community would benefit from having a more narrowed age ranges on the website and the press releases, and I don't disagree. And some folks may remember it was different at the start of the pandemic. We then changed the age ranges to match the state epidemiological report for consistency. I agree it's not great age distributions, but we wanted to be consistent with the state reports, and that's why we did it. I have provided narrower ranges in press releases, and I've done well over 100 interviews in the last 5 months when it was a public health advantage to do so. Most of you may also remember in the first 5 months of the pandemic, I never mentioned a specific town with any cases, but I have in the last 12 days, and I have narrowed the age distribution because there is a public health advantage to do that. We will continue to provide more information as there is a public health advantage to do so, while safeguarding folks' individual privacy to their health condition.
Okay. Yeah.
Somebody said our assisted living centers seem remarkably free of COVID-19 compared to the west side of the state, and they are. Knock on wood. In part, that's because we've been very fortunate. In part because we have an outstanding case investigation contact tracing team within both Whitman County Public Health and WSU. And in part because our owners and managers and operators of our assisted living facilities and long-term care facilities in Whitman County have done an absolutely fantastic job to date. But I've been very impressed working with them. Early in the pandemic, we spoke to them daily, and then we moved to every other day, but we've had very, very frequent conversations with those folks. We help them get PPE. And I will also tell you, because it's been our health department's priority, there has been 3 or 4 times where either a staff member has tested positive or a visitor tested positive after visiting. And we have gone to those facilities in less than 12 hours, and we have taken samples often ourselves because it's a population that has a hard time getting to healthcare. And there are some rapid test machines in the area that we have been able to utilize. Both Pullman Regional Hospital and the Reservations Clinic in Plummer, Idaho, have made those machines available to us, specifically when addressing risk in long-term care and assisted living facilities. And so the few times that we have felt there has been a risk to them, we have gone to them immediately, taken samples, and had results back within 6 hours, while also taking a broader set of tests to run by a traditional PCR to verify that we didn't miss anybody. So we are very aggressive, and we operate very, very quickly if we feel that those institutions are in threat.
So I see
Dr.
two
Gunn?
questions for Dr. Gunn.
Yep.
Yeah. So the first question about the University of Oxford vaccine, and that is another viral vectored vaccine. It's actually a adenovirus one, so it's also expressing the spike protein of the virus. I think that one is looking quite favorable. The only reason I focused on the mRNA ones was actually primarily the announcement yesterday that it looks like the mRNA vaccines are kind of moving forward into phase 3. But I think it is doing quite well, and it's always good just to have multiple vaccines going and effective, so that just in case, the mRNA one doesn't necessarily work for a certain part of the population, other vaccines can help fill that void. Yeah, and I also see a question about the effectiveness of masks, and I think that the sneezing graphic actually really kind of demonstrates sort of how far these droplets can spread, and so anything that you can have in front of your face, it really limits that. Even if some get through, it's going to cut down a vast majority of those droplets that are actually getting spread out. There's actually also been recent, so even beyond sneezing and coughing, even talking loudly is actually enough to, sort of bring out droplets as well. So that's another aspect. So, so even if you're sort of outside without a mask on, if you're yelling, then it might not, you're still sort of generating a lot of aerosols and a lot of virus. So yeah, so I can't recommend masks enough. And then I do hope that, yeah, everybody just, just because if you're, before you're even symptomatic, you may be having virus, you just don't know. So yeah, I would always encourage everybody to err on the side of caution and wear masks.
I think we had one more question in the chat for Troy. Do you need more volunteers for contact tracing?
I appreciate the question and the suggestion that folks may be able and willing to volunteer. There has been a number of folks who have come forward to volunteer of recent, and my days blend together like they do for everybody, but either this morning or yesterday morning at 9 o'clock, I was asked to provide HIPAA training for a group of volunteers that are going to be helping the WSU team. And I said, sure, I'll be on there. It was Zoom, and I thought it was going to be 4 or 5 folks, and it was 16. And so they've really ramped their capacity up a lot. And then we had another group of 14 at WSU who have offered to volunteer that we have put on hold until probably midweek next week to see how it looks with the new testing. As I mentioned, we could do 80 to 100 cases a day now, but it does get fatiguing to do that day after day after day. And of course, that's 7 days a week. And so the reality is we could probably do 180 cases or so a day next week. It just gets to be, depending on how long it goes, how fatigued people get, and we may bring volunteers on later to just kind of be able to roll people off to give them a break. But right now we're doing well. We have a surge capacity, and we also have spoken with Spokane Regional Health District and the State Department of Health. Should for some reason we get 300 or 400 cases in one day, we have outlets for surge capacity through both of those entities. So I appreciate the offer, but right now we are okay for case investigation contact tracing.
Awesome. So I see a question from Barbara about the history of pandemics and what long-term consequences and what kind of ending might we see with COVID, and that's an incredibly good question, and something that we've been thinking about and debating about a lot. And so, I mean, as with with many pandemics, once if we can able to control and sort of get rid of or not basically remove the sort of or reduce the susceptibility of people, then eventually it will, start to subside. Whether or not that takes months is very unlikely, and especially if we get an effective vaccine, then maybe we're looking at about a year or two. I mean, I think that we are let's say for previous pandemics, let's say 1918, we have a very different environment now that compared to what we had previously, and then some other pandemics and plagues have really gone on for like centuries. So I don't think we're necessarily looking at that. In terms for long-term consequences, one thing that I'm particularly worried about is the effects on sort of long-term health for some of the COVID survivors. There's been reports of people that have really decreased lung capacity, severely even after they've cleared virus. We've actually also seen, sort of these long-term consequences with, like, let's say the Ebola epidemic too, where there are these unknown pathologies that are coming out in patients that we think is related to having survived the viral infection, but, we still have to figure out exactly, the best treatments for it. So, I mean, I'm optimistic that with a vaccine that we will see the end, hopefully within, I'm going to go ahead and say a year or two, but of course anything can happen. And of course we must be prepared in the future for, after this virus, there's probably going to be another one. So I think investing in the like sort of the infrastructure, you know, testing, being aware of these kinds of things and being aware that they can come out and emerge into the human population and spread rapidly is, I think that's going to really help us in the future as we tackle whatever comes next.
I'll take the next question about can anyone get a test? The short answer there is no. Where would you go to get a test? Most primary care offices could do the test, and they don't all choose to, but Palouse Medical, Pullman Regional Hospital, and Pullman do most of the test in Pullman if it's not student related. And then if it's student related, you know, it's kind of the 4 entities that I mentioned that will be coming online next week. The longer answer, can anyone get a test? If you're symptomatic, yes, you can get a test. If you've been identified as a close contact of someone who was positive by the health department or one of our agents, you can get a test. If you need a test for travel or some other reason, you can get a test. But if you just, hey, I want to know if I've got it, I want to go in and get a test, we still haven't gotten to that point. And we haven't gotten to that point primarily because there has not been a federal response to this virus to set us up with a national testing protocol and a testing supply line to get us to that point. The second part of that question is how long it take to get results. For most of the 5 months that we've dealt with this pandemic here, we've been fortunate that we've gotten results in 24 to 36 hours. A couple of days ago, I did get a report that we've had a little batch of tests that have taken 72 hours, which is not good. They are speeding up again. And part of that is because almost every test that is done in Pullman up until either the last day or two or in the next day or two, there's a change coming, has gone to Spokane for a session by Insight, and then Insight has redivvied it out, and it possibly could redivvy it out back to the Waddle Lab in Pullman, but there wasn't local accession. Insight is bringing someone down. In fact, I think 2 or 3 folks to be able to do a session here so that the samples taken at Pullman Regional Hospital, Palouse Medical, or WSU can go straight to Waddle, and we'll speed that process up, and we will get back to 24 hours or less. I will tell you, and as that Dr. Gunn's graph showed, because you can be symptomatic, you can spread the disease before you're symptomatic, and it decreases rapidly after you're symptomatic. That makes it critical to do case investigation, contact tracing absolutely as soon as possible. And so when we start seeing labs 3 days or longer, they really get to be where they're almost useless from a public health standpoint. And that's part of the issue the rest of the nation has dealt with. Very few places have had the fast turnaround that we have. And if you're in an area where your tests are taking 5 days or longer, it serves no public health service at all to get the data that long. Troy Henderson, what is your budget status, short and long term, of your office? Well, for folks who may have been following it, I've been here for about 7.5 years, and I have slowly whittled the budget away since I've been here, but through some efficiencies. When I started, there was a very large management team, and we've streamlined that, and we've been stable for the last few years. The commissioners have always been incredibly gracious to me for any monies that I have said I need from local revenues for the health department. And in general, public health in Washington state, the thought is 25% of the funding should come from local taxes, 25% from fees, 25% from the federal government, 25% from state. And that is not where it was when I started. There was a much bigger section from local revenue for whatever reason. We have gotten it shifted down more to kind of 4 quarters. Since COVID has started, there has been a lot of new revenue streams, mostly federal, but it's passed through the state. The reality is for 2020, I'm overwhelmed with more money than I can spend. And in fact, they don't give it to you unless, you can invoice for it. And so I've got more money on the table for public health in Whitman County than I can spend in 2020, which is okay. It'll go back to other public health offices. 2021 is more of a question because, as those of you who have worked in government before know, they rarely want to commit funds in a new budget year. And so 2021 is going to be largely unknown until we probably get into the new budget year for the federal government, which starts October 1. Do I know where the 300 students are isolating dorms? So anytime someone tests positive, we ask them if they're able to quarantine or isolate in their residence. And that kind of depends on who they live with. Do you have an upstairs, downstairs? Do you have more than one bathroom? That kind of thing. You can successfully quarantine and isolate in a home with other folks. WSU has developed isolation quarantine rooms for people who live in the dorms, but they weren't intended for folks who don't live in the dorms. And the 12,000 folks who came back largely have apartments. Kind of the good news early on, when a lot of students in that age group were testing positive, everybody in their apartment was positive. And so if 3 of you are positive in an apartment with 3 folks, you can all quarantine together. So the bulk of those folks are isolating in their own residence. We do have 22 rooms in Pullman that the health department has control over. Community Action Center pays for those through a grant for us that we work with. And we have 4 rooms in Colfax that we can use for folks who need them. We have never been over 50% capacity on our current 26 rooms, but we are looking to increase capacity specific for WSU students, anticipating we will probably see increased numbers next week. And we are trying to work with WSU to open up some of those quarantine isolation rooms in the dorms for folks who live in town.
Awesome. Do we have any more questions, or have we gotten through everybody?
There was just one question at the top about sharing data. Latah County does not share data with Whitman County. Is this true?
No, they share data. As I mentioned, we have good communications for case investigation, contact tracing across the border between Latah County and Whitman County.
Okay.
There was a question about a saliva test that Dr. Gunn could address better than me.
I think the saliva tests are coming online. I think that they're getting better and better. And I guess sort of the question is, I guess sort of like the how specific or sensitive would they need to be, sort of is actually having a positive, even if you have a false positive and then if somebody then quarantines, is that, sufficient to help drive down, the numbers there. That they are coming, they are being developed, they are getting better and better. So hopefully we'll be able to have something soon. Oh, yeah, so there's a final question about the prediction of evolution of the COVID virus. So far, I mean, it's not a rapidly evolving virus. It's sort of evolving at sort of the normal pace that you would expect. There has been one mutation that has been identified that did seem to take over, that was not present initially, that then seemed to take over and was present then in far more infections than the original one that was isolated. So there's sort of there's a hypothesis now that may have increased the infectivity of that particular virus, but the studies are still ongoing and it's still a little bit unclear. But there was certainly a change, and normally when a virus makes a change, it's having some positive impact for the virus. So that is what we've seen so far. But that's only really one mutation that's really taken hold of the millions of cases around the world. So I don't think we're going to necessarily see sort of something super, super fast, not like what we see with influenza.
In regards, are we at risk of being demoted to phase 2? Short answer is yes. As I mentioned, if we start seeing viral spread to the broader community, I would anticipate local authorities, either the Board of Health or the health officer, imposing restrictions. And they may be narrowly imposed to Pullman if that's where most of the viral activity is. They're not likely to say, hey, Pullman's going back to phase 2. They're more likely to do specific actions that they think will help slow the spread of the virus. And it may very well look more like phase 1 than phase 2. I did send a message to the Board of Health and the health officer several days ago, and they're well aware that I'm watching this daily, but I told them that they need to start considering what actions do they think we could take that would make an impact, and start getting your verbiage ready. And the county lawyer was read in on that to make sure that we're ready if we need it. But yeah, if this continues to spread, and especially if we see it getting to the broader community, you should anticipate some restrictions. If I could, I'd like to ask a quick question of Dr. Gunn. you had a slide up where it was tracking the mutations of COVID-19, and through the mutations, you could see it kind of travel from city to city, and there were these lines that showed the travel. I noticed 4 or 5 lines from the United States toward Europe, although it wasn't on the map. I did not notice any lines from the United States heading west. Could you talk to that a little bit?
Yeah, so yeah, unfortunately I was only able just to show the United States data, but actually NextStrain has sort of the global patterns as well, and you can actually look on a date by date. So I will say that if there must have been some cases going back, although I don't know for sure, but we can certainly look on NextStrain to see whether or not that was the case. But
would it be fair to say that a lot of the early transmission into the United States then was out of Europe, as that graph indicated, or would you need to look back at that?
I think so. That was coming from different areas. So there was part that's certainly coming from China, but then there was also one strain that was actually coming in from Canada, another certainly from the New York group that was almost certainly a Europe virus that came in. And so now we're sort of getting this mixing overall. Yeah, I wish I had time to show like a little video, because you can actually track it over time, and you can see sort of like, you know, they have the lineages of where the different country of origin comes from, and it's fascinating and scary at the same time. Actually, I'll put the link in there for you.
Great idea. Do we have any other questions?
It's been very interesting.
It's been a wonderful presentation. Thank you so much. I've learned so much. It's very scary, so I want you all to stay safe and wear your masks.
Yes.
Yeah.
And don't
Totally
go shopping every day.
I would like to comment that if any of you would like to be put on the mailing list, you should contact somebody, look at the website and contact someone to get on the mailing list.
The mailing list for the league.
That is a great point. Yeah, actually, if you like PM any information to one of us, we can make sure that you get put on there.
Because we have some great programs coming up, and you would be able to, you know, find the information easily.
Yes.
Helen, thanks again for setting all of this up. You did a great job.
Excellent.
Thanks.
Yes.
Happy that we got our speakers here. So good.
Thank
Our
you so much for
speakers were
having us.
incredible. Thank you.
Thank you so much for having us. It's really great to speak with you all. And please, if you have any other questions, feel free to email me and get in touch.
Good to know. Way to be on it.
Okay.
All right. Thanks for coming, everybody.
I guess that's the end of our meeting. Thank you.
Bye.
Good night.
Bye-bye.